Okay, let's see if it's recording, because last time I thought it was, yep, it's recording. Okay, good. So the first thing I want to just quickly just review what we've done up until this point on genetics, okay? So let me just clear the canvas here. Okay, so we've been looking at, initially we started with DNA replication, right? Which, if you remember, is you take a strand of DNA and you copy the entire molecule, right? So remember we talked about the way you read it and the way you write it. But the idea is in DNA replication, you make two exact copies. So you end up having an extra copy now of everything, right? Sorry, it should be red. So you end up getting two exact copies of your original DNA molecule. That's DNA replication, right? And that's done through complementary base pairing, right? So if you read A, we build T, 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 right? That's what we talked about a while ago. Then last time we talked about transcription. So in transcription, you take a section of the DNA. So let's say we were to zoom in and take a look at this strand, okay? What you would see, and let's say again, that's 5 prime and that's 3 prime. What you would see is that in here, there are regions that are called genes, right? And when you transcribe, what we mean is we actually take one of those genes and we make a complement copy of it, right? So you take this gene, let's say this sequence, let's call that gene A. Let's say that sequence is A, T. And we know we read from 3 prime to 5 prime, right? Like that. In transcription, you copy the gene by, through complementary base pairing, right? So you would make an RNA molecule that's 5 prime to 3 prime, like this. This is your RNA molecule. And this would be, there's no T in RNA, so this would be U, and that would be A. And that's called transcription, right? And then we talked about translation is when you take this, which is going to eventually become mRNA when it's fully complete, and you're going to convert that into an amino acid list, right? And that is done through translation. These are the three things that we talked about. Okay. Now, before we start, a question about the COVID-19 virus, and just jump in here. There was a question about what the type of virus it is. If the COVID-19 virus was a positive sense or a negative sense virus. Did anyone get a chance to answer that question? So what is meant by positive sense RNA virus? No one got it? So the positive sense RNA virus would be equivalent to the mRNA. Okay, it would look like this. This is your mRNA. All right. The negative sense RNA virus would actually look like the complement of the mRNA that's going to undergo, that's going to be used for translation. So, in other words, the COVID-19 viral genome is equivalent to the mRNA. So, let's say, for example, here we have the mRNA UUUA. So that would directly become then translated, right? If it's a negative sense virus, it would be like the code is AAAT, which then needs to be converted to UUUA and then get translated. So the difference then is this undergoes direct translation, because it's equivalent to the mRNA, whereas this has to be first has to undergo complementary base pairing. Right? And then once it undergoes complementary base pairing, then it becomes basically mRNA equivalent, which leads to translation. Okay, so that's the difference. So so if you come across that question, now you know how to answer it. Okay, so what we're going to do today is we're going to look at the translation aspect, and we're going to be able to read a gene to see, because not all of the gene gets translated. In fact, you have questions about that in your lab on the COVID-19 virus. There are sections of the viral genome that does not get translated. So what we're going to do today is we're going to look at how to read it. Okay. So this is the PowerPoint we're going to look at. Okay. Let me just make this a bit bigger so you can see it. Okay. So this here, ladies, can you see the screen?
Yes.
Yeah. Okay. Good. Good. Because the last time I did a presentation and halfway through the kids told me they couldn't see the screen, which was like I had to redo the whole thing. Okay, so what you're seeing in this picture is actually kind of cool. The here there is a main line that's you can see running down here. This is a strand of DNA, and the branches that are coming off of it, that is actually mRNA. Well, you can't even see that. And then all these little dots, those are ribosomes trying to translate the mRNA into an amino acid sequence. So in this picture here, you're seeing basically the central dogma of genetics. If you remember, that means that you take DNA and you convert it into RNA, and then you convert that into a protein, which is kind of cool because you're seeing that all in this one picture. So today we're going to talk about reading a gene. So here is just a picture of a strand of DNA. Gene could be on either strand, right? So for instance, here is a section of DNA. You can see the double-stranded section, right? The anti-parallel 5 to 3 on one side, and then 3 to 5 on the other side. So you can see that this is the copy of one of these two strands. So it would obviously have to be this strand that was copied, because when you copy the RNA, you make a complement copy, and it's also anti-parallel, right? So the 3' becomes a 5', and the 5' then becomes a 3' on that end. So we're going to talk about how this works. But what I'm going to show you, though, is a gene. So here's actually the gene we're going to read, okay? This gene is actually been trimmed down in size. The reason why it's been trimmed down in size is otherwise we would have to read the actual size of the gene, and genes can be quite big. So like, I don't think you would appreciate if we had to read this. This is quite big, right? So instead of reading something this big, what we're going to do is we're going to read a smaller version so we can fit it on a slide and we can see it nice and easy. Because the bottom line is the parts that are in this smaller sequence are the exact same parts that you would see in the bigger sequence. It's just easier to see. There is a difference, though, in that what we're going to read is smaller because the introns have been removed. Okay, so and that was done because of this process, which I'm going to show you. Okay, so what you're seeing on this slide is transcription, right? You take your gene and you transcribe it through complementary base pairing, and you make mRNA. And if you remember, what happens to the mRNA is you cut it up. That process was called splicing. And that is when you take out the introns, right? You remove the introns. And here you can see, we talked about this, like the introns are this section, that's actually a lowercase. You can see these lowercase letters versus the uppercase letters. So in eukaryotes, the introns actually get cut out, okay? And if you remember, the advantage of doing that is then you can rearrange the exons and produce different combinations. But in any case, you can see that the mature mRNA is smaller because part of it has been cut out, spliced out, right? So then if I said to you, what's the mRNA missing at this point, the mature mRNA, you would say, well, it's missing the introns. Okay, so now if we take that mRNA and we do something called reverse transcription, okay? Now let's think about that term, reverse transcription. That means you're going from RNA to DNA. You're going in the opposite direction. So this actually is something that goes against the central dogma of genetics. If you remember, the central dogma of genetics is you go from DNA to RNA to protein. But sometimes you can actually go from RNA to DNA, is what you're seeing in this on this slide here. And that's because if you remember, the DNA and RNA are complementary to each other. So you can make RNA from DNA, but you can also make DNA from RNA because of complementary base pairing. Retroviruses do this. Negative sense viruses they don't reverse, well, they do have to do sort of like this process in terms of taking their negative sense RNA strand and converting it to a positive sense to complementary base pairing. But the idea is it doesn't have to always go from DNA to RNA. You can see here the opposite process. And retroviruses like HIV will do this to copy their genome because their genome is an RNA genome, which then gets converted into DNA, which then gets inserted into your own genome. So this is reverse transcription. The enzyme that runs this process is called reverse transcriptase. And the name of the enzyme tells you exactly what it does. Now, here's the thing. If we take the RNA that does not have introns, and now you reverse transcribe it into DNA, and then you take that DNA, okay, and you make, okay, sorry, if you look at that DNA, now it's, single-stranded DNA. You know that you can make it into double-stranded DNA, the complementary base pairing. This new section of DNA would be a little bit different than the original because now there's no introns. So because the introns have been cut out, that means that what we're going to look at is smaller. So the actual gene sequence of this gene would be bigger because the introns would have been included. This gene is something called oxytocin, okay? And it's actually quite a small gene. The one on this slide is called ABCD1, which is a much bigger gene. But I said that, reading a smaller gene is easier to read, and it's got the same parts. So whether we read a small gene or a big gene doesn't really make a difference. So let's look at how to read this gene, okay? And just keep this in mind. What we're going to look at when it comes to this gene is something called the coding strand, which is highlighted here, okay? So the coding strand of this gene is the actual strand that looks identical to the mRNA. So it would look something like this, okay? Here is your up top, purple and red, is your double-stranded DNA, okay? Then you can see the white below it is right here. You can see that that is your mRNA, okay? Now, you can see that the mRNA has been copied from the red strand because it's complement to it, right? if there's an A here, you see a U here, right? If there's a T, you see an A. So this mRNA, this mRNA that you're seeing in white came from the red strand, okay? But you'll notice that it's actually identical to the purple strand, okay? It's identical to it. Well, except for, where there's the U, you would have to replace it with the letter T, right? So the red strand, which is what was used to make it, it's called the template strand, okay? The purple strand is called the coding strand, and it is equivalent, or we can say identical, to the mRNA, all right? The reason why I'm telling you this is because the sequence that we're going to look at, this one here, this is the coding strand. So the actual DNA sequence that would have been used in transcription would be complement to what you're seeing here, right? Instead of it being an A, the actual gene that would have been red would have been a T. Instead of it being a C, it would be a G, and that would be a G too, okay? The reason why we're looking at the coding strand is because it is identical to the mRNA, and therefore you're going to see certain features much more easily when we look at the anatomy of the gene, okay? So here we go. So what this gene codes for is two things. One is a hormone. The hormone is called oxytocin, okay? This hormone is used during labor. When a woman is giving birth, she has contractions. This hormone causes contractions, okay? Now, the gene also codes for a transporter, okay? So this gene codes for two things. It codes for the hormone, and it codes for transporter. Okay. Now, we're going to look at the parts of the gene. Now, it says that the gene, or there's a term here called a preprotein, okay, ranges from 37 to 411. So let me look at this. This is I just highlighted this. This is the same as this, okay? I just highlighted it to look at the different sections. So what a preprotein is where we have translation, okay? So a preprotein is everything that would be translated. Now, it says that it starts at position 37, okay? So what I'm going to show you is where position 37 is here. These, if you notice, there's a bit of a separation because they're grouped by 10. So like, for example, this is 10. This is another 10 is 20. That's 30. That's 31, 2, 3, 4, 5, 6. So this right here, this is position 37. It says that the preprotein starts at 37, okay? Now, if you look here, 37 is A. The next letter is T, and the next letter of that is G, all right? A, T, G. Now, what's special about ATG, and you may have noticed this when you're doing your COVID-19 lab, is ATG is an important codon. It is the beginning of translation. Now remember, what we're looking at is the coding strand. So you have to substitute the letter T for a U. So ATG would actually be not ATG as mRNA, it would be AUG. And AUG is methionine, which is the start of translation. And what you should see when you do your COVID-19 lab is that whenever you have a protein being translated, the first codon you'll see is ATG. Again, keep in mind that the T is actually really a U, okay? And you can see that right there. So that's the beginning of translation. It says the preproprotein ends at position 411. 411 would be down here. I already highlighted it. Okay. So 411 is right about here. That's 411. Okay. Here's 421. So this is 411. So you can see that it actually ends right here. This is where it ends. And the next three letters after 411 are T, G, A. Now, why is that important? Well, remember, it's not T, it would be a U. So why is UGA important? Because again, if I look at my genetic code, UGA is actually a stop codon. You can see it right here. So my protein begins here at start and it ends here at stop. So everything from start to here, all of that is my protein. Okay? And it's called a preprotein. You might recall when you did a question about insulin, a pre-pro insulin or pre-pro anything is just the initial protein when it's immature. Okay. Questions so far? You can just jump in if you have any questions. Okay. Now, if you look at the term pre-pro-protein, okay, there are three parts to this. Okay, there is the pre part, there's the pro part, and then there's your protein. Okay, the protein part is the mature protein. The pre-pro protein is the whole thing. Now, we're going to separate the three parts. So the pre part is also called the signal sequence. But I'm actually getting, sorry, I'm getting a little bit ahead of myself. Forgot to mention one thing. So I'm going to backtrack. What is all of this then in front? So I forgot to mention that, which is really, really important because this is also connected to your lab. So if translation starts here at ATG, okay, then that means all of this section in green does not get translated. All right. So what is this? What is this section? What's it called? Does anyone know? Starts with an L.
Leading sequence.
Say it again.
Leading sequence.
Yeah, very good. So yeah, very good. It's the leader sequence. Very good. Now, when you're doing your COVID-19 lab, you might come across a different terminology for it. It's also called the 5' UTR. Okay, the UTR standing for untranslated region. If I look at this image, okay, you'll notice that the so here's my DNA sequence up here, right? You'll notice that not all of it is transcribed, right? So your RNA would start to get transcribed at a particular region after the promoter region, right? If you remember, the RNA polymerase has to find this promoter region to begin transcription, right? So this is your RNA from here to here. But then you can see not all of it gets translated because there is a section in front and there's a section in behind that does not get converted to protein. This is called, you can see it's called a leader sequence. It's also called the 5' UTR. And then this part would, and if you're going to come across this with your COVID-19 lab, this is called the 3' UTR, okay? The untranslated region after the 3' end of your mRNA. Okay, so I forgot to mention this. Sorry. So this section in green is that leader sequence. Okay, sorry about that. I totally forgot about that. So let's go back into that. What is this preproprotein? The preproprotein is everything, but now we're going to separate what is the pre part. Okay, so it says that the pre part is actually something also called the well, it doesn't say it on the slide, but I'm going to tell you. It's something that's actually called the signal sequence. Okay, so this signal sequence, and the signal sequence is actually very easy to find because it starts at the beginning of translation. So it starts here. So it starts right here. So it starts at the beginning of translation, which is where you have ATG, and it ends where the protein begins. Now, how do you know where the protein begins? Well, you won't know. That you'll have to be told. So you'll need a bit of information. So how do we know that the signal sequence, which is also the pre part, ends there? We know where it begins because that's easy. Beginning where translation starts. How do we know where it ends? Okay, you have to be told where it ends. So it says that the protein begins at, I think I have it on, yeah, right here. So the mature protein begins at spot or nucleotide 94, okay? Which means this spot right here, this is position 94. If you do the math, this is 61, that's 71, that's 81, that's 91, this would be 94. So this section here, from start to where the actual mature protein begins, that primary section, that's called the pre section, also known as the signal sequence. Okay? Now, its function is just simply because what actually happens to it is it actually gets cut out. You might recall that from your work with insulin, that the protein undergoes something called proteolytic cleaving or cutting. Now, the reason why it gets cut out is because it's no longer needed. Its function is to serve as an address label. So once the protein knows where it's going, once the cell knows where to send it, then it gets cut out. So it gets discarded. It's kind of like, think of like a letter. When you put a letter on the envelope, you put an address on it and you send it to somebody, right? But then someone receives the letter and they take the letter out of the envelope. They don't keep the envelope, they discard it, right? The address label is no longer important anymore because now the letter has arrived at its position. So this signal sequence, which begins at translation and ends where the mature protein begins, gets discarded once the protein arrives in the cell at the location it's supposed to arrive at. Okay? So that's the pre-section, also called the signal sequence. So if we do the math, then if we take a protein, which is initially a preprotein, and then we cut out the pre, which is the signal sequence, we cut that part out. Then what you're left with is something called, oops, sorry, what was that? Let me do that again. If we cut that out, if we eliminate the pre part, then what you're left with is something called a proprotein. So in this case, the proprotein would be from here, which is this section, right, all the way to down here, where the section just before the stop codon. That's going to be your protein. Okay, and you can see it's missing the pre part because that has been cut out. So from here to here is your proprotein. Now, if we take out the pro part, then what you're left with is just a protein. So if I have a proprotein and then I cut off the pro part, then I'm left with a protein. So what is the pro part of all of this? Well, it says that my mature protein, my mature oxytocin, is from 94 to 120. Okay. Which, by the way, is 27 letters. And if you remember, when translation happens, a codon is read 3 letters at a time, right? So one codon is 3 letters, so 27 letters would be 9 codons. Each codon gives you 1 amino acid. In other words, 27 letters gives you 9 amino acids, okay? So this mature protein is from 94 to 120 in this sequence, which happens to be the part in purple, okay? So if you want to make a protein, if you want to make oxytocin from pro-oxytocin, here's the question. So let's say I want to make, so this we talked about from the part that you cut off the address label to the end of the protein. That's called your proprotein. So in this case, the example would be pro-oxytocin. If this was insulin, it would be called pro-insulin. Okay, so how do we go from pro-oxytocin to oxytocin? What do we need to cut out? What do you think? What has to be cut out?
Protein?
Which one? The pro part or the oxytocin? What do we need to cut out to make pro-oxytocin into oxytocin? What do we have to cut out?
Pro
Yes.
part.
Yeah, pro part. So if oxytocin is from here to here, that means the rest gets what? Cut out. The rest gets cut out. So just to recap, okay? So I know this is a lot. The leader sequence is from the beginning to where translation starts. That's easy to find. From translation starting to translation finishing, that is your pre-pro protein. Okay? The pre part is from the beginning of translation to where your mature protein starts, which is the first part of translation. That's the pre part, also known as the signal sequence. That gets cut out, gone. So we cut out this section. All this gets cut out. Okay? Then you're left with the proprotein from here, in this case, to the end down here. If I cut out everything else, all of this, then what I'm left with is a mature protein. Okay? So these are parts of the gene. We're not done though. We have actually one more part. And you may have noticed this when you were looking at your COVID-19 genome. Did you notice that towards the end of the genome, there was one nucleotide in particular of the 4 that there was a lot of it? What letter was it? Of the 4 letters, right? A, C, G, and T. Which letter had the highest frequency at the end of your genome? Do you remember?
A.
Yeah, very good. A. Now, that's actually really important. If you remember, when mRNA is made, at the end, there is a tail that's attached to it. It's a tail of lots of A's. Okay, it's called a poly A tail. All right, and if you look at your COVID-19 sequence, you'll see that. Now, and the reason why you see this because if the viral genome looks like mRNA, it does. It has a leader sequence that doesn't get copied. It would have, so look, this then, all of this from here to here, right? That is not translated either. So that would be your 3' UTR, right? That's the part that does not get translated because translation stops here. So your COVID-19 viral genome looks like this, and it gets translated into protein. Well, it's not one protein, it's many proteins. But it also at the end has a long, or has quite a bit of A's in it because mRNA gets A added to the end of it. It's a poly A tail. It's added by an enzyme called poly A polymerase, which you can see right here. Okay, poly, and the name tells you exactly what it does. It adds A. Now, how it works is it scans the gene, and it looks for a particular sequence, which is highlighted right here in blue. Okay, it looks for a sequence which is AATAAA. When it finds that sequence, it knows that it has to make a poly A tail. Do you remember what the poly A tail was used for? What its role is in genetics? Why does mRNA get a poly A tail? It was for protection. So the viral genome has a lot of A's at the end, of its sequence because that offers protection to the genome. If it didn't have all those A's, the genome would actually get broken up by enzymes in the cytoplasm. So it learned that trick that mRNA has a poly A tail for protection. So it has really its own poly A tail also to protect it. Now, so the enzyme poly A polymerase looks for the sequence AATAAA, and it knows to make poly A tail. Kind of like the way RNA polymerase looks for a promoter region. What's cool is that you might actually have more than one of these sites. In fact, if I look over here, I highlighted this one, but I could have easily highlighted this, right? Because that's also AATAAA. There could be more than one poly A site for recognition. Okay. So this is the parts of a gene. All right. We went over the leader sequence. So if I erase all of this. So just to quickly recap, we talked about the leader sequence is the first part of the gene. So when we transcribe the gene into RNA, the first part is going to be your leader sequence. And you could see that here. So it's easy to find because it's at the beginning of the RNA and it ends where translation starts. It's also called the 5' UTR, okay? Where translation stops is where you have a stop codon, and everything after that is called the 3' UTR, or the 3' untranslated region, okay? The part that's translated, that's your preprotein. We talked about the first part is your pre part, known as your signal sequence, which is just simply the address label. We know where it starts because it starts at the beginning of translation. The only way you'll know where it ends is you have to be told where the mature protein begins. And it says the mature protein's from here to here. So the proprotein would be from here all the way to the end of translation. And then if you cut out this, you're cutting out the pro section, you're left with the just the protein, in this case, oxytocin. Okay. And we then also looked at how to identify the poly A site. That's it. Those are the parts of a gene. I know that was a lot, but I am recording this lesson. That way you can always just go back if you have any questions about this. So look, I'm going to stop recording, and then if you have any questions, I'll take questions, okay?